Hi, my name is Emma Price and I work in the Neurobiology Research Group based in the Institute of Translational Medicine at the University of Liverpool. I'm going to talk to you today about my PhD project, which is discovering new genetic mechanisms involved in chronic pain associated with ageing. I've just finished the first year of my 3-year project, so first I will give a little bit of background information, discuss our aims and give a quick update on what we have found so far. Finally, I will discuss the research impact of our findings. First, I would like to tell you a little bit about our research group. We mainly study the genetic factors that underlie neurological dysfunction. I personally study the genetic factors underlying chronic pain associated with ageing, whereas other members of the group study different disorders, including Alzheimer's, Parkinson's, motor neuron disease, and healthy ageing. Previously, our lab has also worked on schizophrenia and epilepsy. At the moment, one of our main research areas is investigating the role of mobile DNA elements in neurodegenerative diseases, which I will now give you a brief introduction to. It was once thought that the genome was the same in each cell in the body. However, we now know that there are parts of our DNA that we refer to as jumping genes, and these can change position within the genome. This is a process we call mobilization. We also know that this process occurs in neurogenesis to create what we call somatic variation. The purpose of this is to generate diversity in neurons, and this is what contributes to making each of us unique. Whilst this mechanism is vital during our development, it is also known that this process occurs once again during ageing. If unlucky, jumping genes can potentially insert into parts of our DNA that are crucial for the correct functioning of the cell and cause mutations. This can cause disruption to cellular function and contribute to neurodegeneration. The activity of jumping genes in neurons has already been associated with diseases like schizophrenia and motor neuron disease, and their mechanism has been well studied in the brain. We know a great deal about the activity of jumping genes in the central nervous system. However, we don't know much about them in the peripheral nervous system, and this is what my project is all about. There are 3 main aims to my research. The first question I would like to answer is, are jumping genes active in sensory neurons? If they are, this will contribute to the variation in the peripheral nervous system and ultimately affect how each individual processes external stimuli. The second question I would like to answer is, does the activity of jumping genes occur with ageing? If so, there is more risk of mutations to occur that affect the correct functioning of sensory neurons, and this will disturb sensory pathways. We know that chronic pain associates with ageing, so we are very keen to explore this mechanism as a contributor to this condition. The final question I would like to answer is, what impact does all of this have in the development of chronic pain? For example, in an ageing individual, the increased activity of jumping genes may cause numerous mutations in their peripheral nervous system. The disruption caused to sensory pathways may result in that individual becoming sensitive to certain stimuli that would otherwise not cause pain. This could result in the activation of pain pathways and a state of chronic pain. The ways in which we are going to explore the mechanisms of jumping genes in the peripheral nervous system is to create a model using sensory neurons grown in culture. The type of sensory neuron we will use is called the dorsal root ganglia, which I will refer to as DRG. These cells harbour crucial pain pathways to detect harmful stimuli. We will first measure the activity of jumping genes in the DRG, using molecular biology techniques to detect their presence. We will then conduct further experiments using DRG from different age groups to measure if the activity of jumping genes increases with ageing. Finally, we will measure the impact of jumping genes on DRG function. For example, how do aging DRG with high jumping gene activity respond to painful stimuli in comparison to younger DRG with limited jumping gene activity? To conduct the experiments, we will mainly use molecular biology techniques that focus on DNA analysis. You may have seen recently in the media a new technique called CRISPR, which enables precision editing of the DNA inside cells. Using this tool, we are going to remove a jumping gene that is predicted to affect the function of a key pain gene in the DRG and assess the impact on cellular response. The experiments we plan to conduct will enable us to answer our questions about the activity of jumping genes in the peripheral nervous system. In addition to the experiment being conducted in our lab at the University of Liverpool, we also have several ongoing collaborations to expand and assist our research. We are working with Dr. Susanna Helias at the University of Pech, who has trained me extensively in the process of isolating DRGs from the spinal column, how to culture them appropriately, and how to extract DNA and proteins for further analysis in our lab at Liverpool. I spent 2 weeks in Hungary working alongside her team to learn this delicate process, and we hope to continue this collaboration throughout my project. To further our work using DRG, Dr. Bernard Frank at the Walton Centre has enabled us to have a collaboration in order to obtain DRG, and this has been crucial in the creation of a protocol for the use of human tissue in pain research at the University of Liverpool. We are currently working with Dr. Dan Carr at the Wolfson Centre for Personalised Medicine to study the variation of jumping genes in patients diagnosed with osteoarthritis. In a study where patients were treated with tramadol and had various success rates, we hope to identify patients that have certain variations in their genome that may potentially be used as biomarkers to identify patients in the future that will respond well to the treatment. And finally, we have a collaboration with Dr. Mike Iadarola at the National Institute of Health in America. Mike has set up a pain genes database which can be used to search for gene activity in sensory neurons. His team has kindly agreed to assess the activity of jumping genes in these sensory neurons, and this will contribute valuable data to our model of chronic pain. I have just finished the first year of my project, and I am pleased to announce that we have confirmed that jumping genes are active in sensory neurons. From the collaboration with Pech University, we have measured the activity of jumping genes in DRG and have confirmed their presence within the cell. This is an exciting finding as it has not yet been discovered in the peripheral nervous system. I will now spend the 2 remaining years of my project to address the final 2 questions. Does the activity increase with aging, and what impact does this have on neuronal function? So what does all this mean for the patients? Firstly, if our model is correct and an increase in jumping genes does occur with aging, the increase in somatic mutation may disrupt how sensory neurons respond to stimuli and contribute towards the development of chronic pain. Secondly, this will ultimately change the perspective on how peripheral nervous systems function as we age. This will influence future research and the management of chronic pain. Trials are currently underway to suppress the activity of jumping genes in motor neuron disease and cancer. Therefore, there is opportunity to limit damage associated with aging of sensory neurons. And finally, the completion of this project will establish a resource at the University of Liverpool to study neurological disease in human tissue. This will enable the validation of pain models, explore further therapeutics, and collaborations to expand pain research in the future. I would like to thank my supervisors, Professor John Quinn, Dr. Jill Bubb, and Dr. Bernard Frank, for their continuous guidance whilst conducting my research. My colleagues who have contributed valuable knowledge and assistance whilst conducting experiments in the lab. Our collaborators who have progressed my learning and offered great contributions to our project. And finally, to the Pain Relief Foundation for their support and funding. Without them, this research would not be possible. Thank you for listening, and if you have any questions, please feel free to email me at the address provided.